Welcome. Today's video it is regarding the polymerase chain reaction. You call it as an PCR, which is the in vitro amplification of the DNA. And this technique it has been first proposed by the Gobind Khurana when he was working on the chemical synthesis of the gene. But this technique was not work out at that time because there is no sequencing technique was available. Also, there is a no thermostable DNA polymerase is available, and when there has to be the amplification of the DNA, there is a need of the primer. And to design the primer, there is a need of the DNA sequencing. So this technique it has been forgotten, and after 15 years later, it has been came in the mind of the Kary Mullis in 1985, and then the PCR it has came into the scientific world. Now this polymerase chain reaction which has been given by the Kary Mullis, this technique it is involving this technique it is involving the three steps. First one it is a denaturation, second it is the annealing, and the third one it is the extension. So we are going towards what principle and the steps of the PCR, and here we have the double stranded DNA. So when it is a double stranded DNA, it has to first get the single stranded, that is, it has to be denatured. Why it has to be denatured? Because when you are going to amplify the DNA, when you have to amplify the DNA, the primer it has to bound with the DNA, and if it is previously bound with the second DNA, then this primer it cannot anneal, it cannot bind with the template DNA. So the first step it is a denaturation, that is a separation of the strands of the DNA, that is there is a melting of the DNA, and it has been carried out at a higher temperature, and that temperature it is a 94°C. This temperature it may vary according to the GC content of the DNA. If GC content it is a higher one, then the melting temperature, that is the denaturation temperature, it is also going to be higher one. Okay, next step it is the annealing. After denaturation, after separation of the two strands of the DNA, then the primers now they are allowed to get annealed, allowed to get hybridized with the template DNA. Okay, this it is a yellow one, it is the template DNA, and this one they are the two primers. Now here the temperature it is a 55°C. Here also this temperature it may vary. How? According to the melting temperature of the DNA. Okay, that is the TM of the DNA. And this annealing temperature, this annealing temperature it has been set accordingly the TM. And how it has been set? 2 to 3 degrees lower than the melting temperature of the DNA it has been set. Okay, so these two primers they are going to specifically bind to the template DNA. Here it is very important that the primers that are used, that are going to be hybridized, that are going to bound with this template DNA, they should be specifically binding. So here this annealing temperature it is very important. Okay, this annealing temperature which has been set accordingly the melting temperature TM of the DNA, these primers they are going to specifically bound with the template DNA. So there is the chances of the false positive results they are very less when annealing temperature it has been set properly. So these primers they are going to hybridize with the template DNA, and after that the extension it is going to be extension it is going to be there, and that extension it will be at a 72°C. At the extension, the DNA polymerases, the thermostable DNA polymerases, it is allowing the extension of the primer. That is, there will be the polymerization of the DNA. The primers which has been annealed, to these primers there will be the addition of the nucleotides. So as the nucleotides they are going to be added, the daughter DNA strand it is going to be synthesized. Okay, so here you can see, here you can see after completion of the extension you are going to find out that to this template DNA the daughter strands they are synthesized. Now now if you can see here, okay, the template strand, it is a larger one, and a daughter strand, it is a smaller one. So when you are going to amplify the DNA, if you are taking the genomic DNA, the size of the genome from that genomic DNA, a small specific region you have to amplify. So these primers, okay, so these primers only going to bound at a specific regions. So these are the regions which are not required to be amplified. Okay, so these are the regions which are okay, these are the regions which are not going to be amplified. The primers, they are specifically binding in between the DNA, and they are going to amplify only the some part of the DNA, and that it is of about the 1 kb of the DNA, it is going to be amplified. But here we have to amplify the DNA from 1 nanogram to the micrograms. So there is a need of repeating of this cycle. Okay, the repetition of the cycle, that is the denaturation, annealing, and the extension. These cycles, it has to be repeated. That is you have to set the number of cycles, and most of the times the cycles which has been set, they are about the 30 cycles. So the 30 times these reactions, these three steps, they are going to be there, and then you are going to find out that the small amount, that is of about 1 picogram or the 10 nanogram of the DNA, it has been now amplified to the micrograms. Okay, next and the final step, it is the final extension. In the exams, what you do, you write the paper, and in last 10 minutes or in the last moment, what you do, you check out whole the paper, and you find out that each and every question, it has been written, something you have kept, that you are going to complete. Okay, so that completion, that left out, it is going to be completed into this final extension. Okay, so this final extension is carried out at the 72 degrees Celsius for the 1 minute. So these are the steps of the PCR. This it is the principle of the PCR, that is the PCR, it is going to occur in the three steps. It is a denaturation, annealing, and the extension. Now here, parameters which are to be kept in the mind, that is the parameters affecting the reaction or the essential components of the PCR. Which are the essential components? What is required by the PCR to carry out the PCR? What is the most important things which has to be kept into your appendix or in your microcentrifuge tube? That it is the first one. It is you are requiring the thermostable DNA polymerases. When Kary Mullis, he has first invented the PCR, he has used the enzyme, and that enzyme, it was the clinofragment. Okay, the clinofragment, which has been derived from the DNA polymerases 1, and DNA polymerases 1, it was from the E. coli, and E. coli, it was a mesophilic organism. So the so when there is a denaturation step, when the temperature, it has been raised, at that time what is happening, the enzyme, it is denaturing because it was unstable at a higher temperature. So after every cycle, the alicot of the enzyme, it has to be added to the reaction mixture. So 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 here, to have the automation of the PCR, there is a need of the enzyme which can tolerate the higher temperature. That is, there is a need of the thermostable DNA polymerases, and the first thermostable DNA polymerases which has been found out, it was a tag DNA polymerases from the Thermus aquaticus, and this it is the most commonly used enzyme in the PCR. Okay, which is that it is a tag DNA polymerases, and this tag DNA polymerases, it is used in the 0.5 to 2.5 units per 25 to 50 microliter of the reaction. If you have the 50 microliter or you have the 25 microliter of the reaction mixture, final reaction mixture, at that time you have to use the 0.5 to 2.5 units of the enzymes. And most important, what is required? The primers. Okay, what is required? The primers, they are required. Why the primers are required? Because DNA polymerases by itself, it cannot start the polymerization. Okay, and for that purpose, what is required? The primers, they are required. Primers of the length 17 to 20 nucleotides it is required. And what was the concentration? It is a 0.1 to 0.5 micromole of the primers it is used per reaction. And next, what is important for the polymerization? What is required? DNTPs. Okay, for the extension of the primer, okay, for the extension of the primer, the nucleotides it has to be added, DNTPs it has to be added. So there is a need of the deoxynucleoside triphosphate, that is there is a need of the DNTPs. Which are that DNTPs? DATP, DTTP, DCTP and DGTP. And these four, okay, that you are going to use, they should be in an equal concentration, that is equimolar concentration it has to be used. Otherwise there will be the errors into the amplified DNA. And the concentration which has to be used, it is of about the 200 to 250 micromole. Most important thing into the PCR, it is what? In the reaction mixture, it is a divalent cations. This stacked DNA polymerases, it is requiring the cofactor, and here it is a Mg++, that is the magnesium it is required. Some of the time magnesium it can be replaced by the Mn++, and it is in the 1.5 millimolar. And the buffer which is used, it is a TCl of the pH 8.3 to 8.8 at which has been set at a 20 °C, and its concentration it is a 10 millimolar. Now, when you are preparing the buffer at room temperature, at that time its pH it is alkaline. Don't forget that the pH it is affected by the temperature. So when there is a raise into the temperature, when there is a raise into the temperature, that is from the 20 °C, when it is when it is a 72 °C, at that time the pH it is going to drop, and it is dropping at the 7.2. So here, when you are going to set up the pH of the buffer, it is 8.3 to 8.8, so that when there is a actual polymerization temperature, at that time it is going to be a pH 7.2. Next, what is required? The monovalent cations they are required, that is the KCl it is required, the potassium it is required. Next, there is a need of the template. Without template, there is a no amplification. So what is required? The template it is required. And which types of the template it can be used for the PCR? It may be the single stranded DNA, it may be a double stranded DNA, it may be a closed circular DNA. When you are going to find out the efficiency of the PCR, it is a lesser to the closed circular DNA than the linear one. Now, when you are using the concentration of the template DNA, for the mammalian, it is of about the 1 microgram it is used, in the yeast 10 nanogram, for the bacteria 1 nanogram, and for the plasmids it is a 1 picogram. So such a small amount of the template DNA it is required for the PCR, which is going to amplify it up to the micrograms of the DNA. I think you may have understood the basic reaction of the PCR components which are required for the PCR. If you have liked my video, please subscribe my channel and also share my videos with your friends, so the knowledge it is going to spread. Please write down the comments into the comment box. If you have any doubts, if you have any questions, you can write down into the comment box. I will answer your questions. Okay, thank you.